Hello, this is Miss Dagenford, and we're going to be examining our synapses today, and that should get us a nice, interesting topic of discussion because now we get to look at how things piece together. We've talked about the structure of a neuron. We've talked about how neurons then conduct impulses. We have talked about how we can make them faster. And now we're going to look at how they then send the signal to the next neuron. By the end of this video, you should be very comfortable with the parts of a synapse. You should be comfortable with how a synaptic transmission occurs. You should be familiar with what a neurotransmitter does and then how they work together. So we're going to try and cover quite a bit. So let's go back to our original drawing where we had two neurons together, and they end with these lovely little axon terminals, and they're going to connect to another neuron, or they could connect to a muscle. They could connect to the neurons in our brain, or to the spinal cord, but the idea is that they are going to send a signal somehow. Now, this signal is not going to be an electrical signal, and here's where it gets a little interesting. It's going to be converted to a chemical signal. So let's dive right in. Well, this picture that you see is actually coming from right here, because what you have is the presynaptic neuron, and then the postsynaptic neuron would be after this gap. So what are we looking at? We are seeing the action potential travel down the axon. So here's our little action potential, and it's going to occur here, and then it's going to occur here, it's going to occur here. It's going to be traveling down, and it hits the end. Now, as it hits the end, it's going to signal a couple of events. And so let's go ahead and jot these down. So we have an action potential traveling down the axon, and as it hits the axon terminals, it's going to signal a couple of things. First, it's going to signal the calcium channels. Now, we talked about calcium as being a really important molecule here for signaling. Calcium channels open, and then therefore calcium diffuses inside. And then here's a big point here. We have synaptic vesicles. And what they do is, and this is a nice review for the cells, this is an exocytosis, where the synaptic vesicles fuse with the membrane, and as they fuse, they release those chemicals called neurotransmitters. And neurotransmitters are going to diffuse across this gap, the synaptic gap. Some books will call it a cleft. So here we have the synaptic gap or synaptic cleft. So they'll diffuse across, and then they will connect to a receptor. Now here you should start feeling a little more comfortable with this material because we just talked about how signals can cause things to happen. So it's going to fit this receptor, it's going to cause this receptor to open, and it's going to cause ions to flow in and out. And depending on the receptor and depending on the signal, it can cause the postsynaptic neuron to either get closer to threshold or farther from threshold. So that is an exocytosis. The vesicles are pulled to the membrane, and then they fuse, and then they dump out the neurotransmitters. And just as a nice reminder, guess what little molecule is bringing them down on big microtubule tracks? You'll recall this from the beginning of the year, our lovely little motor protein that is bringing our synaptic vesicles, and they're walking, quote unquote, walking down the microtubules, and that's how they get there. And for some neurons, this can take days because they're down the length of your leg or your arm, and so traveling down that can take a long time. Now, why do we care about this? What's going on? Well, First, we care about this because this is happening in our brain. These signals are telling our brain to eat. We're hungry. Ooh, chocolate is good. We really like chocolate. Or, coffee. Coffee is good. We like our coffee. And so these are what's causing us to react the way they do. This is a big implicator in clinical depression. What's happening to our synapses, and how many neurotransmitters are we sending across the gap? This is unfortunately also why illegal drugs can cause the problems that they do. You've heard about the epidemic of heroin overdoses, and we know that most of the illegal drugs, well, they work based on how they interact with those receptors, causing our neurons to go overboard or quieter, whether or not they're suppressants or depressants, uppers or downers, things like that. So let's take a look at how we can put this together. So we have our poor person stepping on a Lego, and that's going to be our stimulus. And then, of course, eventually we're going to say ouch. But how do we get to the point where we say ouch, and then how do we pull our leg back? Well, let's go ahead and label that. That's our stimulus, the pain. It's going to be perceived and then sent by a sensory neuron. And the signal is going to, here's our little dendrites, and then the pain signal is going to be sent up to our spinal cord. And so I'm going to go ahead and here, I'm going to go ahead and finish my drawing. This would be my spinal cord, and it kind of has a little shape inside here, which would be like the little bundles of cell bodies. What happens in the spinal cord? It would be the meeting of those synapses, of the neurons, of the synapses. Now, what's really wild is think of how complex it can become very quickly. So, for instance, not only do we have the motor neuron that it's going to connect to, so let's go ahead and do that first. You step on the Lego, the signal travels up to the spinal cord, and it immediately synapses to a motor neuron, which loops back to the quadriceps. Now, here I'm going to take a little bit of artistic license. This is going to be the quadriceps, and of course other muscles involved, but let's just focus on one. So here we'd have our quadriceps, which would be telling our leg to lift up. Well, how do we get the quadriceps to lift up? Well, we get a signal from our motor neuron that's going to that's looping back, and then it's going to tell the quads to contract. You pull your leg up, and at least you've removed your foot from that Lego. But at the same time that you had the synapse here, as it was connecting to the motor neuron, you had another neuron that is traveling up to our brain, and the signal as it gets to the brain, that's going to lead us to cringe, make facial expressions, think of all the connections that can happen, cry out in pain, hopefully not curse. Now, what's interesting is, though, we can't override this. This is a reflex. A reflex is involuntary. A reflex is a very simple pathway, and it really is a stimulus response pathway. It's as simple as that. Something happened, it connects to a motor neuron, it causes a motor neuron to send a signal, a muscle or a gland will then do whatever it needs to do. But we can't override some of these reflexes. Breathing is a reflex, but we can be told to breathe at the doctor's office. Heart rate, to a large extent, is involuntary, but we can also calm it down if we also control our breathing rate. And in the same way, if you're holding a baby, so maybe the big brother spilled his Legos and you're holding his little brother in your arms, and as you're walking across the floor you step on the Lego, well, you're going to override that stimulus to hop around in protecting that baby. So it's a really neat thing to start thinking of the layers of decisions that happen in our brain. But at its very simplest, here's our stimulus, our sensory signal, synapse, motor neuron, and then this is called an effector. And the effector can be either a muscle, in this case, or it could be a gland. Now, we long ago in August, we talked about Pavlov's experiment with dog and salivary glands, and the same thing can happen with us. We just don't notice it. If you walk into the dining hall and you smell pizza, unwittingly your salivary glands are prepping you and secreting saliva. Now, hopefully you're not drooling, but you're producing saliva. So there's your nice little introduction to reflex. But at the same time, we can also send inhibitory signals as well as excitatory. So let's take a look at a few circumstances and clean this up a little bit. And again, you'll have a sense of just how amazingly complicated this can be. So we've got two neurons, the green and then the red. The green and then the other green. And these are, think about stoplights. The green is go and then the red is stop. Now, if we focus on this first green one on the top, the graph below is basically saying that these are two signals, the first signal and then the second signal. But notice what happened. They're so far apart that the resting potential is reached before the other signal happens. So there is no signal. We have not hit threshold. But let's look at case number 2. We're getting two signals, but notice the frequency has increased. So now we get a second signal before it has reached resting potential. This is called temporal summation. We get to sum the signals because we've not let this drift back down. And notice now it hits threshold, you're going to get an action potential. The more frequent that signal is, the stronger that impulse is going to feel, even though the action potential is the same. If we keep sending and keep sending and keep sending, we're going to get that sensation that this is a very strong impulse. The next one is going to be two neurons. So here we have the first neuron and then the second neuron, but they're both sending at the same time. Now, because these are two different neurons, we call this spatial summation. We had temporal summation, now we have spatial summation. And these so far have all been excitatory, but take a look, you can also have inhibitory. The red one here, this is an inhibitory signal. And notice, because we are taking it away from threshold, we're not reaching that point and we're not getting an action potential. Well, hope that helps. We're going to really cover these and make sure we understand these before moving on, but this gets us started. Have a lovely day.